Good afternoon. It's about 2:45, and what we want to do is look at another section of Chapter 3. I've put Chapter 1, Part 1 in to your week 3, and Chapter 3, Part 2. So we're doing Part 3 at this point. I'll have one short Part 4, hopefully maybe tomorrow morning for you. Anyway, what we want to do is look on page 87 of your textbook. Now, what we just finished with was the plasma membrane. You remember that. So here we have on page 87, down at the bottom, you have the organelles, those little tiny they are microbiological, but they're not germs, but they are micro in size, and they carry out all these little functions in our cells. They call them machines. And machines, of course, are sometimes very simple, sometimes very complicated. But as you look on the second column, you see, or actually it's really the first column, you see they're called little machines, and they compartmentalize part of the cytosol. So they carry out functions and activities that if they were out in the cytosol, they might damage some other structures. We're talking about having enzymes and some acids, because some of the enzymes work at a good pH of 5 or something like that, because they're made to work at that pH, but they could damage things. So you look at the bottom of that column, it says organelles compartmentalized by membranes are often called membrane bounded. That's fine. Now, as you look over on page 88, let me get my clamp on here, and you got a great picture of a cell. So you want to know the components of that cell, and you can go back and refer to them, and we've also got them in other pictures that we'll look at in a minute. So you look over to the left column, the first column says membrane organelles include the mitochondria. We're not going to study peroxisome, but we are going to study the endoplasmic reticulum and the Golgi apparatus, and also the lysosome. We just can't learn everything. There is a course called cell biology, and so some of you may have had that somewhere else, and it's amazing. It's a wonderful course. It makes you appreciate what you're made out of. Anyway, those are the organelles that we're going to be looking at, and so as you look at the second paragraph, it says each type of membrane enclosed organelle has a membrane that is unique in terms of its thickness and composition. And so it says in spite of those differences, an organelle's membrane is still similar to the plasma membrane. So we've just got a variation of the plasma membrane inside the cell around a number of what we call organelles. So a lot of things go on, and we want to look at them. I'm not going to get in there too deep because it's not a cell biology course. But as you look over on page 89, you see mitochondria. And you look at the second line there, it says it's involved in energy production. You probably knew that if you took Bio 110, or if you took Biology 101 in another place or whatever, you probably picked that knowledge up. And you see it says the main source of energy for cells is chemical energy, and we get that from producing ATP. We turn glucose and fructose into ATP so that we can drive the machinery. You know, you got to have gasoline in your car if you're going to go. So we have to have this ATP. And you look at the next to the last line, it says most of our ATP is produced within the mitochondria. So they can call it the power plant of the cell, and that's fine. Notice on the second column, mitochondria can be in great numbers in cells. And you see in the third line down, it says, for example, cells such as the liver, which metabolizes a lot, that requires a lot of energy. So you got to have mitochondria to produce ATP. Skeletal muscles, I mean, we're working those to some degree all day long. And some of us like to go out for exercise. So got to have some more of those mitochondria in the muscles to produce the ATP. Cardiac muscle Can't stop, can it? No, it can't stop. So you've got to have those mitochondria. Now notice on the second column on page 89, you see this second paragraph. And I want you to read that. It talks about how each mitochondrion has its own DNA and enzymes for protein synthesis. Now, as you read through here, you see it also says it has its own ribosomes. So it's actually carrying out, making its own proteins that are special for its function. And if you come on down, you'll see it says that most of the proteins come from DNA that were from the cell's nucleus. But then you see it says the remainder, mere 14, that's what the mitochondria produces. So as you look at the picture down here, you see you've got an outer membrane, and you come down a little bit on figure 316, inner mitochondrial membrane. So it's a double membrane structure, got some ribosomes as you see there. Now that's about as far as we're going to go with that. Except one more thing. One more thing. As you look on page 90, and you come down the first column in the second paragraph, you see it says the mitochondrial matrix. You remember what that is? It's not a movie, okay? It's material, got a lot of liquid in it, probably, proteins and so forth. And you see it says contains enzymes and proteins that break down organic fuels. What's an example of an organic fuel for us humans? Suppose I ask you that on the test. What would you put? Okay? So we're going to skip over to the second column now, ribosomes. You come down to the bottom and you see that, and you see there are non-membrane enclosed organelles. They're called an organelle, and we typically think of an organelle as being surrounded by a modified plasma membrane, but in this case, they're not. And you can see from the top of the page, next page, you got a couple of components that come together like a hamburger. And you see that as you look at that little paragraph, each subunit is made of a complex assembly of ribosomal proteins and a type of ribosomal RNA. They are valuable to us. Look at the bottom paragraph. Two types, free and bound. The free ribosomes, you see there, top of the next page, says they're suspended in the cytoplasm, where they make proteins that are used in the cytosol. You can't just turn any protein loose in the cytosol. It could create damage, so you got to make some special proteins. Cells got to know how to do that. How does it know how to do that? Just sit back in awe as you read this and go, golly, I never knew that. Then it says bound ribosomes are associated with the membranes of other cellular structures. Look at the next sentence. All ribosomes start out free, but some will attach themselves to the endoplasmic reticulum of the nuclear envelope or the nuclear envelope. And then the bound ribosomes typically make proteins that are going to be exported from the cell, transported in certain organelles like lysosomes, or in some sort of membrane, inserted into maybe the membrane of the cell. When they make proteins, you remember those proteins that were on the surface of the plasma membrane? Where'd they come from? Well, we're going to be finding out about that. This is just a little taste of it. We've got a little bit of complex stuff at the end here, but we're not going to get to that today. So anyway, you want to read that and remember that. Come over to where you see endoplasmic reticulum. This is another structure that's got a modified plasma membrane. And so they sometimes call it, as you see on page 91, the ER. Let me get my clamp in place. And it is continuous with the nuclear membrane. So there's a reason for that. It's just not there for the fun of it. There's a reason there. And notice as you come down, it says current evidence. Now that means we don't have it all figured out. But that's part of the fun. There are a lot of people that like to do research and they study stuff and then they find out the answer. And, oh, whoa, look at that. We discovered that. What happened there? So we know a little bit. You're going to know a little bit. You're going to know a little bit enough to make a living as a dentist, a doctor, nurse practitioner, radiologist, person, whatever. But you come on down and you see where it says this space contains enzymes. That's the space in the endoplasmic reticulum. Go back and take a look at it, or you can look over on page 93 and look at the space in there. Got a lot of space inside, and there are enzymes there. And you see it says that those enzymes carry out a variety of reactions. Notice in the next couple of sentences, it says the rough endoplasmic reticulum, where the membrane is covered, is in bound ribosomes, and the smooth endoplasmic reticulum, or smooth ER, that doesn't have any. As you look back, you'll see some of the rough of the endoplasmic reticulum has no little dots on it. Those dots would be the ribosomes. Those that have the ribosome on it, they are that part of the ER is called the rough ER. Sometimes they call it granular. Now, rough endoplasmic reticulum. Recall that the proteins must be folded into their proper shape. So when a protein is made, and we have not gotten to that point yet, just having a chain of amino acids is only the primary structure. There are other things that have to happen. But if you see under rough endoplasmic reticulum, the ribosomes, the site of protein synthesis, only assemble the acids into a polymer called a polypeptide. That's a chain of amino acids. Come down to the next paragraph, the polypeptides synthesized on bound ribosomes pass through the ER into the inside, into the lumen, where enzymes catalyze reactions that fold those chains into a particular shape to do a particular job. And you and I don't have anything to do but just eat. Go ahead and get some good barbecue and turn that into broken down amino acids that these guys can put it together for you. Produce more proteins to carry out the functions of our body. I want you to look on the fifth line, 1, 2, 3, 4, 5, in the middle of that column. And it says the rough ER, RER, recognizes proteins that have folded into an incorrect shape. How in the world do they do that? And then what they do is they break that down and start all over again. You have to say, how in the world do they know how to do that? Well, that's kind of like spell check on your computer, isn't it? How'd spell check get on your computer? Just happen to show up, right? Anyway, keep that in mind. That's why I love this stuff. Every semester I get excited about this. Look at the second column. Many of the proteins that enter the rough ER are folded. To be folded are secretory proteins destined to be exported from the cell. Most of those proteins are glycoproteins. Remember how we're going back now to the beginning of the chapter? Glycoproteins. And the rough ER enzymes catalyze reactions that attach sugars, that's your glyco, to certain amino acids, and you get glycoproteins. And then it says when the secretory protein is fully assembled, it leaves packaged in a small membrane transport vesicle. And then it's sent to the Golgi apparatus, which we'll mention in just a second. And it's put outside on the outside of the cell, exocytosis like we talked about, and you end up getting that protein in the area where it needs to be. Just fascinating. So that's where those proteins come from. The glycoproteins come from the rough ER. Come down to the smooth endoplasmic reticulum. Notice in the second line it says no role in protein synthesis. But here's what it does. Calcium storage. We always think of calcium with bones and teeth. It's got other functions that we need to learn about a little bit later. Storage of calcium ions. Detoxification. This is your smooth ER. And if you take in some things, it'll detoxify them. Toxic, like a person with alcohol. And nothing wrong with a glass of wine or beer or whatever, just in moderation. But if somebody drinks a lot, the smooth endoplasmic reticulum is going to grow, trying to keep the poison from damaging the person's system. So there's the second function. The third one, down at the bottom, lipid synthesis. You see it says the SER, smooth endoplasmic reticulum, synthesizes the bulk of the cell's lipid membrane components, phospholipids. So there you have, the inside of the cell has got stuff going on to make the outside perfect. So we have a selectively permeable membrane as opposed to something where everything can go in and out as it pleases. That's probably where something like Lipitor slows down the lipid synthesis. That's why a lot of people are on atorvastatin, or they just call it Lipitor. So it probably works there in the liver, on the smooth ER, and it slows down the making of cholesterol, and down comes somebody's level of cholesterol. They say it does a lot of other good things for you too. My doctor put me on it when I hit 50. That was 23 years ago. Let's go down the bottom of that column and you see Golgi Apparatus. Here's the next organelle. And you see it's located between the rough endoplasmic reticulum and the plasma membrane. You can see a view of that over on page 93. This green structure, it's not green in the cell. They just use colors because we like colors. So there's that green structure that's called the Golgi Apparatus. There's an electron micrograph, 83,000 times magnified. And then here's the illustration. So as you come back to the Golgi Apparatus on page 92 down at the bottom, you see it consists of a group of flattened membranes filled with enzymes. Those enzymes are going to do something special. And they got some other things in there too. And you see it says within the Golgi Apparatus, proteins and lipids produced by the ER get modified. That means maybe the shape is changed to some degree. They get sorted and they get packaged. And then you find that it goes to various places around the cell. Look at the last sentence in the first paragraph. Each sac of the Golgi Apparatus contains different enzymes. How in the world do they know to get certain enzymes? We don't have all the answers, but that's part of the fun. So enjoy it. Now come on down to lysosomes. When you see lyso, think of Lysol. Lysol splits bacteria up, okay? And lys means to split, and some means a body. So a lysosome is a body that's got some enzymes in it that's going to split something up. Might be bacteria, might be virus, or whatever. But you see, it says all cells have to be able to digest nutrients. Even worn out cells, as you see there. And cells do wear out, and we repair them, and then we multiply those cells, and we get them back up to where they should be, so we carry on reasonably well. Lysosomes in that fourth or fifth line, membrane enclosed, contain water and enzymes called acid hydrolases. Now remember, if you see ASE on the end, that's an enzyme, isn't it? Hydro is a root word for hydrogen or water, okay? So you got some hydrolases there that like to function in an acid pH. What would be an acid pH? Well, you read a little further in here, and I think it'll say something around, the second paragraph down there, about the fourth line, it talks about how the pH in the lysosome is acid. Why doesn't that denature the proteins? Because those proteins are made to work at a pH of 5. How do our cells know to produce that stuff? Think about your stomach. Your stomach pH is anywhere from 1 to 2. And we produce pepsin, which works on proteins that we put into our stomach. And that doesn't bother that pepsin because it's made a particular way to function at a particular pH. All we got to do is put some steak in there, put some barbecue in there. Okay, those hydrolases, those would be proteins, wouldn't they? Because enzymes we're going to say enzymes are always proteins. Let's see what else we can do. Okay, now you know a little bit about the Golgi apparatus. So let's go over to page 96. So we've covered the lysosome, the Golgi apparatus, and the endoplasmic reticulum. Now, page 96, got just a little bit of stuff here to give to you. Module 3.5, the cytoskeleton. The cell has a skeleton just like we did. We've already mentioned this, if I remember correctly. And as you come down and you look at the first paragraph, first column, first paragraph under there, and you see it says the components of the cytoskeleton function as a cell's highway. Things move along these skeletal features, these structures. Did you ever get to see Willy Wonka's chocolate factory? And there were little buckets going across on these cables and stuff. Well, our cell is something like that. So the cytoskeleton has a number of functions, and one of them is to be the highway where organelles may be transferred from one part of the cell to another, dropped off, how they know to let go. We'll never find it all out, but that's part of the fun. So we come down to the second paragraph. Cytoskeleton is made up of several types of filaments, protein filaments. There are just two functions I want you to know about it. There are others, but we're just going to get two. It gives the cell its characteristic shape and size. It helps it keep a particular shape, and that actually goes for the ER too. And it helps to keep that lumen open, and the Golgi apparatus and so forth. And then it also functions, you look down at the bottom of that column, and it talks about movement. Notice it says the protein filaments of the cytoskeleton are often associated with motor proteins that work like molecular propellers. You guys ever been in a boat, and you look over the back of the stern of the boat, and you pull the motor up, and there are these propellers. We have them too. And you see down there, motor proteins are like propellers, and they use energy derived from ATP. Come up to the top of the next page, or the next column, it says the cytoskeleton filaments and their motor proteins are responsible for moving organelles. We've already talked about that. But also notice it's valuable for splitting the cell during division. Okay, keep it in mind, we'll get there. Now, as you come on past this next feature of performing specialized functions, don't get into that. We just can't do it all. But come down to it says in this module, we'll examine filaments that make up the skeleton. We're also going to look at the structure and function of microvilli and cilia and flagella. Now, you probably know a little bit about some of those things. We'll just mention them and we'll be through with these things. Actin filaments. Now, if you've had 110, you probably remember actin and myosin. If you will come down to the second paragraph under actin filaments, type of protein, slender protein, plays a role in moving the cell. If you come to the second line and go to the right, when myosin is involved, it says it's incapable of moving the entire cell. So, you already know that probably about contraction of muscle. Actin is involved in movement. Going to skip intermediate filaments. I want you to come over to page 97 and look at microtubules and centrioles. What I'm interested in is the second paragraph, just a little bit about it. These, again, are components of the cytoskeleton. Microtubules have a wide range of functions. If you look over, they maintain the internal architecture of the cell. In fact, if microtubules are disrupted, the endoplasmic reticulum collapses. Remember that endoplasmic reticulum is doubled, and you got a space in there with a lot of enzymes, but if those skeletons break, just like our skull could be crushed and everything just gets squished. Notice also it says, and the sacs of the Golgi apparatus. Okay, that's one of the functions of these microtubules. Now, finally, look at the next paragraph, starts with in addition, come down to the last 2 or 3 lines. Finally, microtubules form the core of cellular extensions, cilia and flagellum. We're going to talk about that in just a second. Before we get there, though, look at the next paragraph, figure 322. And you see it's talking about a region in the cell called the centrosome. Just flip over there for a second and look at the cell, figure 322 on page 98. And you see this little cloudy area. And it says it's a region of a cell, and it is consists of a gel matrix with a lot of proteins in it. Now, in that centrosome, come to the next paragraph. The centrosome contains a pair of tube-shaped structures called the centrioles. And come down 5 lines from the top there, from the top of that little paragraph. 1, 2, 3, 4, 5, and you see the centrioles play a critical role in cell division. Now you look at those centrioles there. You say, how in the blazes do they play a role in cell division? Pull the chromosomes apart, snap them apart, can bring in the cell membrane so you can seal it off and have a good barrier there. All you got to do is give it some energy. It'll do it. Now, we're going to leave that. We're going to leave those centrioles, but you want to know what they're involved in, cell division. And you see cellular extensions on page 98. And we've got a couple of those we want to mention. Microvilli. Come down 5 lines from the first of that paragraph of microvilli. Cells that are specialized for absorption. That's their function, to absorb things, such as those in the kidney and the small intestine. Have plasma membrane with a huge surface area. And what makes that a surface area so large is you look at figure 323, and you see it's got microvilli. It looks like a bunch of little fingers sticking up. Imagine if everything was flat. You would not have nearly the surface area you would have with all these little villi that are involved in absorbing our food when it is digested. It goes, if you read through there, it's 30 to 40 times more surface area than if you just had a flat surface. So microvilli are found in your small intestine, and they absorb your food after it's digested, small enough to get through the cell membranes. And then you got your kidney for reabsorption. Kidney's an amazing thing. Not the easiest thing to understand, but you'll get into 211 and you'll be awed again as you look at the kidney and all the things it does to keep us in fluid and electrolyte balance. Okay, let's, as you look at the second column on page 98, look at that last little sentence. Actin filaments are found at the core of microvilli. They give them some support, like a little rod going out. And then we have cilia and flagella. Let's see, we're at almost 30 minutes now. I'm going to knock it off about 35 or 40, I think. So cilia, those are extensions also, and they're not the same thing as microvilli. Now, what I want you to do is I want you to read through that section. Cilia actually move. They move material out of our respiratory tract. They're in other places in the body too that we'll get to a little bit later. But they are not like microvilli set up to absorb everything, give you a big area for absorption of food. So they differ structurally and they differ functionally. Now, let's come down to the second paragraph, and I want you to come down to, now I want you to come down to the last paragraph and come to line 2. You're in the first column, last paragraph of 99, second sentence. Cilia are shorter and move in a coordinated beating. They are present in large numbers in certain parts of our bodies. Now, if you look at the last sentence, the cells that line our respiratory tract sweep mucus out all day long. You and I are breathing in dust particles. We're breathing in some pollution. We're breathing in bacteria and some virus to some degree, maybe mold spores or whatever. And they get caught in the mucus in your respiratory tract, and your cilia are always bringing it up. This is why the flu is so dangerous. The flu destroys the cilia. And then the mucus doesn't come out. That means the bacteria don't come out. And they keep multiplying. And it's possible for you to go from a viral bronchitis to a bacterial pneumonia. You don't get the bacteria out of it. It's called a flushing mechanism. When you and I think flush, we think hit the handle and stuff's out of the house, right? Well, that's kind of what the cilia do. They flush the respiratory tract. It's in other places also. We'll get to a little bit later, but right now that's enough for you to know. So anything you put into your lungs, like vaping or cigarettes or whatever, you breathe in some kind of organic solvent or whatever, it can damage your respiratory tract. And then you don't get all that mucus out, and pretty soon you just start having recurring infections. And then the last thing over here in terms of these moving parts, you look on page 100, got a nice little picture of some sperm, and the flagella is the structure. Still has that actin filament in the middle of that one little tail. That's the only cell in the body of humans that has a flagella to propel it. Now, let's do page 100 and 103, and we'll be out of here. So we got the nucleus, module 3.6. You want to check off the learning objectives 1, 2, and 4. 1, 2, and 4. And as you come into the paragraph, you see the bold print term nucleus, and you keep on reading. We discussed earlier the nucleus houses the DNA, which codes for our proteins, our genetic makeup. And then you keep reading. It says the plans, called genes, which is a section of DNA, within DNA are used by different kinds of ribonucleic acid to build our proteins. I'm going to make another video about this. It gets a little heavy in there in terms of what happens, but I'll try to make it as straightforward as I can. So you see the nucleus up top. You see it's labeled. You can see the nucleolus in the center, and you see the nucleoplasm containing chromatin. Now, when you think of chromatin, you think of chromosomes, but the chromosomes is a stretched out chromosome. They're stretched out, being used to produce proteins until they're ready to divide, and then they condense into proteins that you saw and probably saw in the lab when you looked at mitosis and you saw those little finger-like structures in there. Maybe they talked to you about sister chromosomes or sister chromatids or something like that, and the centromere and so forth. That's good. Okay, I'm not even going to mention that because you probably already have had that. Now, you know about the nucleolus and you know the nucleoplasm and so forth. And as the last sentence in the first column says, the most notable component is our DNA. And it's in the form of chromatin until it's time to condense the proteins, and they have reproduced. You went from 46 to 92, and now you're going to pop them apart, and half of them go this way, half of them go this way. Now you got 2 cells with 46 chromosomes, or 23 pairs. The nuclear envelope, that's the membrane outside. Sometimes people call it the nuclear membrane. And you see in there, it says like the mitochondrial membrane, it's a double membrane. And notice it's continuous with the ER. And over here on page 102, you see chromatin and chromosome. You certainly want to know the terms. The chromatin is the stuff that's stretched out. The chromosomes are condensed for reproduction. And you see whenever they have reproduced, and you probably learned this in a lab, you look over here on the second column on page 102, and you come down to the second paragraph, and come down to the third line. It says, during cell division, the chromatin threads coil tightly and produce chromosomes. And when they do that, you say, oh, they've divided to some degree. You got this connector in the center, and that's a centromere, which you see in the next paragraph. A couple of things here. The last thing on page 103, you can read about homologous chromosomes. You know that you're half your dad and half your mom. And if you've got kids, your kids are half you and half your husband. So you can do the quick check, you can know that, and then you come to the nucleoli. You see the word nucleolus in there, dark staining structure. And notice what it says about it when you come down to line 4. We think of a nucleolus as a ribosome factory. Got to have some ribosomes for protein synthesis. And you see the nucleolus is not a membrane. It's not covered by a membrane, but it's a big bunch, an aggregate, as they would say, of proteins, DNA, and RNA. Come on down just after that RNA, come down 1, 2, 3, and it says, however, in the nucleolus, we find segments of DNA containing genes that don't code for a protein, but instead specify the nucleotide sequence of an RNA strand. We have 3 kinds of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA, which we'll get into, as you see on page 103 on the second column, protein synthesis. So I'll try to go over that maybe this afternoon, and maybe I'll be able to put it into your pocket come tomorrow morning. Okay, y'all have a good evening. I'm going to go back to work and see if I can work on this and make it, what would you say? Let's see, there we go. Try to make it as clear as I can. See you later.